Good morning to our audience, and thank you for joining us this morning for the webinar on alternative approaches to manipulating muscle growth and physiology in the yard. I'm Dr. Zeno Hubbard, ruminant technical expert at dsm-firmenich, and I will be your moderator today. Please note this webinar will be recorded and shared with all registrants, and that you can ask questions using the Q&A button at the top of your screen. All attendees today are anonymous, but we do ask that when you submit a question, please note your name will become visible with your question unless you submit that anonymously. Dr. Johnson will address as many questions following the webinar. Now I'd like to present to you Dr. Johnson, Brad Johnson, today's presenter. Dr. Johnson is currently Gordon W. Davis Regent's Chair in Meat Science and Muscle Biology, as well as Professor at Texas Tech University. Originally from Millbank, South Dakota, he obtained his bachelor's degree from South Dakota State University and also holds both a master's and doctorate degree in animal science from the University of Minnesota. Dr. Johnson's laboratory at Texas Tech University focuses on research to further the understanding of skeletal muscle growth, regulation and development of beef cattle. His group has used exogenous growth promotants like steroidal implants and beta-adrenergic agonists as models to study the mechanism of postnatal muscle growth in meat animals. And they've also been using the beef on dairy model to study some of these differences in growth response to implants. Additionally, Dr. Johnson's research team is also investigating novel means to enhance marbling in beef, focus on the receptors present on intramuscular adipocytes, which appears to have positive effects on lipid accumulation. We know that carcass weight gain is one of the main drivers of profitable feedlotting, but new nutritional technologies to significantly manipulate skeletal muscle growth are limited. As a result, nutritionists today are re-evaluating the fundamentals of ruminant nutrition, assessing essential nutrient supplementation, as well as optimizing the use of available growth enhancement technologies, in addition to further refining management practices. Dr. Johnson's work provides an invaluable contribution to an industry always striving for that additional pound of hot carcass weight gain. With that, I'll hand the floor over to Dr. Johnson.
Well, good morning, everyone, and I want to thank Dr. Zeno Hubbard for that introduction and the rest of her team at DSM for hosting this very important, invaluable webinar this morning. So, as the title implies, most of what we're going to visit about this morning are potential alternatives, alternative approaches to increasing or improving skeletal muscle growth of cattle in the feed yard, and ultimately improving that hot carcass weight that Dr. Hubbard mentioned in the introduction. Today, with unprecedented cattle prices, as she said, a 2 to 3 pound increase in hot carcass weight could easily be worth $10 per head. So we're going to address some alternatives to that. To kind of set the table for that, I am actually going to use two very well-known growth promoting strategies, as Dr. Hubbard mentioned in her introduction of me. We're going to talk about steroidal implants, the active ingredient anabolic steroids that make up steroidal implants, as well as beta ligands or beta-adrenergic agonists that are orally active growth promotants that have been used in feedlot cattle growth and development since the early 2000s. So these are going to become the model for which we talk about the mode of action of how these two well-established growth promotants impact beef cattle growth and development in the feed yard. And then we're going to look at alternatives that really use the same mode of action and talk about the opportunities of those to actually enhance carcass gain or carcass weight improvements in feedlot cattle. So as a bit of an introduction, we know that in the United States, in our cattle feeding industry, we're noted for several things. And first off, we would be one of the most efficient producer of beef per animal, for example, of anybody globally. And part of this comes about due to selection of genetics over time for improved growth rates or average daily gain. And at the same time, we're improving growth rates. We know that we also are very efficient in converting dry matter intake or feed nutrients into live body gain or carcass gain. So our feed conversions are some of the best, again, in the world as well. And part of that, again, is coming about by the use of some of these growth promotants like steroidal implants and beta adrenergic agonists that we've already talked about. At the same time, on the other side of the coin, we're also noted for extremely high beef quality or product quality. And part of this is coming from an improvement in marbling scores, the intramuscular fat that we find underneath within the muscle bundles, within the juxtaposed to the muscle fibers, and also due to selection of certain genetics of cattle and manipulating days on feed, we know that we produce a very tender product. But one thing that I want you to keep in mind, and a theme throughout this morning's webinar, is that everything we do to support growth, positive growth rates and efficient growth, often have an antagonistic effect on meat quality. Okay, so we're always doing this balancing act of we want very efficient growth and high rates of growth, but we have to balance that with maintaining adequate marbling and maintaining a very tender product. And so one thing you're going to learn is that we can really manipulate growth and accelerate the rate of muscle growth in feedlot cattle, but oftentimes that's going to have a detrimental effect on meat quality. So we have to balance these things as we try to achieve both excellent growth and efficiency of growth and maintaining high marbling and a very tender product. As Dr. Hubbard mentioned, for many years, we've had the ability to use steroidal implants. And in fact, we've been using steroidal implants since 1956. So the U.S. Food and Drug Administration approved Synovex S in 1956, and we still have that implant today, 69 years later, to improve efficiency of growth and average daily gain and also carcass growth characteristics. Just recently, within the last 2 years, the U.S. Food and Drug Administration has kind of changed how we can potentially use these implants, and specifically how we can use multiple implants on the same animal. So part of Guidance 191 document that came out over 2 years ago, we now have limits of how many implants we can use in a given production phase. And the U.S. Food and Drug Administration would recognize growing and finishing as one production phase. And so you can see at the bottom of this slide, there's only one brand of implants, the Zoetis' line of Synovex implants, that have been approved for reimplanting in this grower finishing phase. And so because of this, we've been kind of limited in the number of implants and maybe the types of implants that we can administer during this grow finishing phase. And because of that, I think that really leads us into why we're doing the webinar today, is that are there alternatives that may come in and offset the need for some of these reimplant programs in cattle while maintaining our typical production responses and efficiency of production and maintaining a real high quality product. And so the outline that we're going to use this morning is I want to first give you kind of an overview of postnatal skeletal muscle growth, because as Dr. Hubbard alluded to, we're going to focus on carcass gain and carcass growth this morning. And so a large portion of that is obviously skeletal muscle, the edible lean tissue in a beef carcass. And so we're going to kind of give a quick overview of some of the challenges and limitations of postnatal skeletal muscle growth. As we start talking about factors that impact skeletal muscle growth, you're going to learn very quickly there's these very specialized cells called muscle satellite cells that become critical at supporting postnatal muscle growth, and in fact are the rate limiting step of postnatal muscle growth. And so we're going to talk about factors that impact the activation of these satellite cells. And in fact, the anabolic steroids, the active ingredients in steroidal implants, we have shown to be crucial activators of satellite cells. So that's going to lead us to talk about the mode of action of steroid implants and working through these critical satellite cells. We're then going to switch gears and talk about a very important enzyme system in all cells of mammalian bodies called the AMPK system. And in fact, the other class of growth promotants that we use in the beef feedlot industry in the United States, beta adrenergic agonists or beta ligands, we have shown to be that are strong activators of this AMPK system in skeletal muscle. And we're going to talk about the implications of activating this AMPK system in skeletal muscle from affecting things like protein synthesis and protein degradation as it relates to beta ligands or beta adrenergic agonists. But then we're going to give other examples of some alternatives to beta agonists or beta ligands that will also activate this same system. We're then going to kind of end the webinar this morning by talking about some true alternatives. And I've kind of just highlighted this on my outline as vitamins. And so we're going to talk about things like vitamin D3 and vitamin A and some B vitamins like biotin that have been shown to work through the mode of action of doing the two things above that I've already talked about. Things like activating satellite cells and also perturbing or positively impacting the AMPK system. And then finally, I'll end with a few concluding remarks from this morning's webinar. So let's move into postnatal skeletal muscle hypertrophy. And as this title implies, we're talking about the growth of muscle tissue after birth. And so to lead off, I think it's important that everybody understands some very key characteristics of postnatal skeletal muscle tissue in all animals, including birds or poultry species that we may deal with from an agricultural standpoint. One of the main key characteristics I want everybody to understand is that the number of muscle fibers is essentially fixed at birth. And so what a muscle fiber is, or a myofiber, this is actually individual muscle cells. So the individual muscle cells that make up skeletal muscle tissue in our meat animals, including avian species like poultry, the number of these fibers or the number of these cells is fixed at birth. And we're never going to change that. And so this kind of limits the ability to grow the muscle because the number of cells is fixed at birth. So if you picture an 80-pound calf at birth compared to now a 1,600 or 1,700-pound market animal in the United States, at harvest, they're going to have the same number of muscle cells or muscle fibers at harvest as the day they were born, okay? So the other thing about muscle fibers is these are some of the largest cells in our body. And because they're so large, they're multinucleated. As you can see on the picture here of this slide, there are many nuclei that spread across this individual myotube or muscle fiber. And that is because they're large cells, those nuclei contain the DNA, which ultimately gets transcribed into messenger RNA, and then that messenger RNA is translated into new protein through protein synthesis to support the growth or the hypertrophy of those existing fibers. So in fact, the amount of DNA or the nuclei number really becomes a rate limiting step for postnatal muscle hypertrophy. And the reason for that is not only are these cells, the fibers are fixed at birth, once the nuclei go into these existing fibers, they can no longer divide. We say they're postmitotic. And so we have to bring in new DNA or new cells from the outside to actually support this postnatal muscle hypertrophy. And we know that the DNA content of skeletal muscle increases dramatically early in life postnatally as muscle is growing very rapidly and very efficiently. And so the source of this DNA or the source of these cells, we now know to be specialized cells called muscle satellite cells. And these cells are very unique in that they lie outside of the cell membrane of the muscle fiber, which we call the sarcolemma, but underneath the loose connective tissue surrounding the muscle fiber called the basal lamina. And the important thing about satellite cells is that they maintain the ability to still divide or proliferate or go through mitosis. And so certain signals will turn these cells on so they can divide faster. And ultimately, after they've divided a few rounds of proliferation, they ultimately want to fuse into the fiber that they're associated. So they lie juxtaposed to individual muscle fibers. And so after these cells have divided, they're actually going to fuse through the cell membrane and donate themselves, which is a source of DNA or nuclei, to that existing fiber. So in fact, because the number of muscle fibers is fixed at birth, we know we have to increase nuclei or DNA content to support postnatal muscle hypertrophy. We now know that muscle satellite cells are the critical rate limiting step of this. And to show this on a cartoon diagram, you can see an individual muscle fiber on the top side of the slide. And the small purple circles associated with that are those muscle satellite cells that are still outside the muscle fiber. And again, the critical thing is they're able to proliferate or divide many times. But ultimately, after they divide a few times, if you kind of look at the muscle fiber on the bottom of the slide, you see more little black dots within that, and the fiber got larger. And so these cells ultimately fuse through the muscle cell membrane, the sarcolemma, donated their DNA or nuclei to the existing fiber, and caused more efficient muscle growth of that fiber, really through the process of protein accretion. So kind of in the center right side of the slide, I show that protein accretion is equal to protein synthesis minus protein degradation. So the rate at which we produce new protein, but we're always breaking down protein in skeletal muscle, but hopefully the rate of synthesis is greater than degradation, and we're going to accumulate more protein mass. So how do growth promotants fit, established growth promotants fit into affecting skeletal muscle growth? And so for the first example, I'm going to use anabolic steroid implants as an example of how this can manipulate lean tissue deposition or carcass gain in feedlot cattle. And so over 30 years ago, we were the first group, part of my master's work at the University of Minnesota, we were the first group to research a combined trenbolone acetate estradiol 17 beta implant called Revelor S. That was the first approved combined TBA-E2 implant in the United States, marketed by Merck Animal Health. Back then it was Herx through Cell. And we did a series of serial harvest studies where we implanted yearling steers with this Revelor S, 120 milligrams of trenbolone acetate and 24 milligrams of estradiol 17 beta compared to a non-implanted steer model. And what you can see from this graph is that looking at carcass protein accumulation, we saw this rather dramatic increase in carcass protein accumulation the first 40 days after we administered the Revelor S implant, or the combined TBA-E2 implant, to yearling steers. So that by day 40 in our serial harvest study, we saw about a 15% increase in carcass protein mass in the carcasses of these steers that had been implanted with TBA-E2 implants. So we were interested in knowing, before I go further, if you look at the lines after day 40 are parallel, right? So they maintain that carcass protein mass difference all the way out to the end of the feeding period of 143 days. But really, most of that increase came about the first 40 days, and then the animals were able to maintain that. So we were really interested in knowing what was going on the first 40 days after we administered the TBA-E2 implant that could be causing this increase in skeletal muscle growth and differentiation. And so in fact, we did some follow-up work where we isolated muscle satellite cells that I talked about on the previous slides from either implanted steers using the same Revelor S implant, the TBA-E2 implant that we talked about in the previous work, compared to non-implanted animals. So we actually implanted these steers, and then we focused in on from about day 35 to day 42, that period of time where we saw this increase in carcass protein accumulation in our original serial harvest study. And interestingly, what we found was when we isolated satellite cells from steers that had been implanted with TBA-E2 implants, we actually saw that those satellite cells were more vibrant and proliferative in a cell culture model. We talked about them engaging in the cell cycle or proliferation with shorter lag phases. So they attached to the dish in our cell culture model and started proliferating much quicker, as this bar graph shows, compared to non-implanted steers, or cells isolated from non-implanted steers. So this led us to believe that, in fact, the active ingredients in the Revelor S implant, trenbolone acetate and estradiol 17 beta, somehow were activating or causing activation of these satellite cells and led to greater proliferation rates, which ultimately led to more DNA being accumulated into the muscle fibers of these yearling steers. And that's really what accounted for this 15% improvement in carcass protein accumulation at the end of the feeding period. In addition to the work we did with the satellite cells early on in the early 1990s, one thing we always see when we administer a TBA-E2 implant to cattle, we always see a marked increase in a very important growth factor called IGF-1, insulin-like growth factor 1. We'll see an increase in circulating IGF-1, and we believe most of that IGF-1 in circulation, that change is coming from the liver. But we also see a change in local muscle production of IGF-1 caused by the TBA-E2 implant. And IGF-1 is so critical at not only stimulating these satellite cells and supporting satellite cell proliferation and differentiation in the existing fibers, but IGF-1 also is critically important at stimulating protein synthesis of muscle tissue and reducing protein degradation. We now know that this increase in protein synthesis is actually coming about through perpetrations through a very important pathway called the mTOR pathway in skeletal muscle. And mTOR pathway has been showing to ultimately improve protein synthesis and at the same time reduce protein degradation. So whenever we alter IGF-1 at the muscle level, we're going to have positive impacts at stimulating protein synthesis and reducing protein degradation. We're going to come back later and talk about other alternative molecules that have been showing to affect not only IGF-1, but also the mTOR pathway. So now we're going to switch gears just a bit. And so I've been talking generally about factors affecting postnatal muscle hypertrophy. And I've talked about how important protein synthesis or protein accretion really is, and that protein synthesis and protein degradation, the balance of those two, cause changes in protein accretion. We know that skeletal muscle, when you take away the water, protein is the number one nutrient in skeletal muscle. And in fact, the most abundant protein in skeletal muscle is myosin. The second most abundant protein is actin. And we know that myosin and actin interact during muscle contraction. The functional purpose of muscle is to contract, and myosin becomes critical, as in fact, it's a hexameric enzyme. It has enzymatic activity to cleave ATP into ADP and inorganic phosphate for muscle contraction. So in beef cattle and bovine skeletal muscle, we have three predominant types of myosin heavy chain isoforms. We have myosin heavy chain type 1, myosin heavy chain type 2A, and myosin heavy chain type 2X. And this is very similar to what we as humans would also have in our skeletal muscle. And so what's interesting is the different isoforms of these myosin, the different myosin isoforms cause differences in growth potential, efficiency of growth, but also from a meat quality perspective. And we're going to kind of go through and talk about each of these. What's important is that we have shown that certain growth promotants can cause the transition of these different myosin isoforms within cattle muscle. And now we're going to switch gears and talk about beta ligands or beta adrenergic agonists. So items like ractopamine, zolpadorol, and lababagron that are orally active mixed in cattle feeds, we can actually show that by feeding these beta ligands, we're going to not only alter the different types of myosin heavy chains, but also cause an increase in muscle hypertrophy or fiber hypertrophy of these different types of myosin heavy chain isoforms. So our lab group for the last 12 or 13 years, I've done a lot of work at looking at different factors that not only impact the type of myosin heavy chain isoforms that we'll find in beef cattle muscle, but also the hypertrophy. And as you can see on this slide, there's 4 different panels. These are cross-sectional areas of muscle bundles. We can do muscle biopsies of the longissimus muscle or the bicep femoris shoot side at the feedlot, and then we can fix these. We fix these samples, take very thin cross-sectional sections of those and put them on a glass slide. And so what I want to point out from this slide is you see 3 distinct different colors. Okay, so the green outline, we use an antibody for a protein that's embedded in the cell membrane or the sarcolemma of muscle to show individual muscle fibers. So you're going to see that we have some of the smaller fibers on these slides are stained red. And these are actually the myosin heavy chain type 1 isoforms. And so you'll know that they're stained red, but they're also the smallest diameter. Myosin heavy chain type 1 are called oxidative fibers. These are aerobic fibers. They can burn lipid or fatty acids as a fuel source, and in fact become very important from an endurance standpoint, say in human performance. The fibers that you see that are kind of yellow or orangish, these are the intermediate fibers. These are the myosin heavy chain type 2A fibers, and truly they're intermediates. They can go back to becoming oxidative type 1 fibers, or they can move forward and become the third type of fiber that I show on this slide, the ones that we don't stain, the ones that you see are black. These are the myosin heavy chain type 2X fibers. And so these are the glycolytic fibers. They're going to use glucose through anaerobic metabolism, produce lactic acid as a product of the metabolism. And so what you'll note from this slide is that the largest cross-sectional area or diameter is actually the myosin heavy chain 2X, the glycolytic fibers. And in fact, when we feed a beta ligand like ractopamine, lababagron, or zolpadorol, we actually are going to increase the size of all fibers, but we're really going to increase the size of these type 2X fibers. So from a meat quality perspective, the only way we can have marbling is we have to have the myosin heavy chain type 1 fibers, the oxidative fibers. And if we have animals that are exclusively type 2X, and we have that in cattle, in double muscled breeds like Piedmontese and Belgian Blue, we'll have no marbling because the marbling is only associated with the oxidative fibers. The other thing that you'll see is a stark difference in cross-sectional area of those 3 different fiber types. Larger cross-sectional area usually results in more efficient, massive muscle growth. But that comes at a cost, as I said early on in my introduction. That increase in cross-sectional area generally increases toughness or reduces tenderness of muscle tissue if we have high levels of type 2X fibers. So we're always balancing at a fiber level where we're trying to maintain marbling and tenderness, but yet we want very efficient, rapid muscle growth. I led into this talking about one thing that we've noticed is that, in fact, beta agonists or beta ligands have been showing not only to transition the muscle fiber type towards a more glycolytic fiber, but we're actually going to increase the cross-sectional area and length of all fibers, regardless if they're myosin heavy chain type 1, 2A, or type 2X, by feeding a beta ligand. And here's some work we did with zilpaterol when we were feeding zilpaterol from 2006 through 2013 in the United States. And you can see that we actually transitioned in the semitendinosus muscle of the hindquarter of these steers towards more type 2X fibers. But when we fed the beta agonist, the beta 2 adrenergic agonist zilpaterol, we actually increased the cross-sectional area of all the fibers due to feeding a beta ligand. And so we had done many studies where we showed that by feeding orally active beta ligand to cattle, we were going to stimulate postnatal muscle hypertrophy, namely through protein synthesis and the protein degradation pathway. And so we were interested in knowing kind of the mechanism of what was going on. And there's a very important enzyme, as I let off in my outline, called AMPK, adenosine monophosphate activated protein kinase. Every cell in our body is going to have this enzyme. And it's a very central cellular energy regulating enzyme in every cell of every tissue in mammalian animals. So what, as the name implies, adenosine monophosphate activated protein kinase, AMP, adenosine monophosphate, is really the product of taking ATP and using ATP as an energy source in cells of our body. And if AMP levels are high, that implies that we've burned all the ATP, right? And ATP levels are probably low. And so the activation of this enzyme is going to start trying to conserve energy. And in fact, it's been showing that when we activate AMPK in skeletal muscle, we are actually going to regulate the fiber types that we find in muscle. And so again, using the beta ligand model, we knew that by feeding a beta ligand like ractopamine, zilpaterol, or levodopa, we're going to increase the rate of protein synthesis of muscle, and we're going to reduce protein degradation. The net effect is we're going to increase skeletal muscle hypertrophy of those individual fibers that I showed you on our histographs. So we were interested early on in knowing what was the mode of action of this? What was ultimately causing a shift in fiber type away from oxidative or red fibers that are so important for meat quality towards a more efficient, larger glycolytic fibers that are important from increasing a carcass lean tissue deposition or carcass gain response. And so some of the early work we did with zilpaterol, we showed that by feeding Zilmax for 20 days with a 3-day withdrawal, we actually increased the abundance of this AMPK enzyme, both at the mRNA level, the gene expression level, as well as the relative abundance of the actual protein. And what was interesting is by increasing AMPK, we actually saw an increase in myosin heavy chain 2X. Those were those big black fibers that I showed on my histograph that are glycolytic, large, very efficient fibers, but lower meat quality standards. But look at the middle panel on the bottom of this slide, myosin heavy chain type 1, which are the red fibers. We actually reduce the expression of those when we increase the myosin heavy chain type 2X. So we felt that the AMPK pathway, as some earlier work by Dr. Gerard's group at Purdue showed in pigs, if we perturb this AMPK pathway, we're going to have this shift from an oxidative fiber towards a glycolytic fiber. And so to do follow-up and further mechanistic work, we employed a cell culture model where in fact you can see that we saw this dose-dependent response. When we added zilpaterol to cultured muscle cells, we saw an increase in the amount of AMPK, and not only the parent AMPK, but for this enzyme to be active itself, which kinases are enzymes that phosphorylate other proteins, it itself has to get phosphorylated to get turned on. And so not only was the native protein increased, but when we did a dose titration study in a cell culture model, we also saw an increase in the phosphorylation of the AMPK. The net effect again was that we altered the myosin heavy chain isoforms that were being expressed away from oxidative towards a more glycolytic myosin heavy chain type 2X. So again, this is kind of a great model of how beta agonists work. And so at this point, before we go on any further, I talked about steroidal implants impacting satellite cells. Interestingly, we know we feed beta ligands the last 30 or 40 or 56 days of the feeding period, typically in the United States. We see these profound effects on protein synthesis, increases in protein synthesis, and reductions in protein degradation. But during that time, we rarely see any more changes in satellite cell activity. So it becomes very important if we want to get the optimal or really the maximal response out of our beta ligands, we got to make sure there's enough satellite cells already in that fiber prior to feeding the beta ligands, because beta ligands are going to have no impact at stimulating these satellite cells. And so that's where the use of implants or other technologies, maybe some alternative technologies at stimulating these satellite cells in the fiber prior to the introduction of the beta ligands at the end of the feeding period becomes so critical. So what other things? So kind of the theme of my talk this morning is really looking at alternatives of affecting some of these pathways. And so interestingly, if you go into the literature, what are some other items that are alternatives that have been showing to impact this AMPK enzyme, very similar to what beta ligands might do? And in fact, a B vitamin, biotin, has been shown to modulate AMPK pathway in a similar way to beta ligands. We know biotin is a natural B vitamin, right? And here's an alternative method of modulating AMPK. But of course, we know in ruminants like cattle, we would have to rumen protect that biotin to get it to the small intestine to be absorbed at a higher level in order to modulate this AMPK pathway. Another naturally occurring substance during corn processing of say, white corn for masa production for human consumption reasons, one of the byproducts of that is ferulic acid. So this would be a phytogenic compound from corn that has been shown also to perturb or alter the AMPK pathway in a similar way to beta ligands. And in fact, some Mexico research has shown that ferulic acid feeding has been shown to really replace zilpaterol feeding in Mexico feedlots. A couple other things I want to talk about before moving into other vitamins. We also know, as the theme of my webinar this morning, alternative methods to improve, say, lean tissue deposition in beef carcasses. We've shown, some of our work in our lab has shown that certain amino acids can also impact this mTOR pathway and satellite cell activity and ultimately affect lean tissue deposition. We've done a lot of work with ruminally protected methionine, and we've also done some work with another derivative of amino acids, hydroxymethylbutyrate. So specifically with rumen protected methionine, we showed that by supplementing 8 grams of metabolizable methionine to feedlot cattle, we actually showed an increase in the abundance of satellite cell activity. And in fact, on this table, you can see that 8 grams of rumen-protected methionine led to increases in myonuclei. And this was brought about by changes in some of these important transcription factors like MIF5 and PAX7 that are important markers of satellite cells. And we, in fact, turn these satellite cells on by providing more methionine. And in fact, we've linked that back to the mTOR pathway that I talked about earlier in my presentation. So let's switch gears and talk about the last 2 items that I want to mention in this morning's webinar are going to be vitamin D, vitamin D3 specifically, and its receptors, and then we'll kind of end with some discussion on vitamin A. But first, let's talk about vitamin D, because I think there's such great opportunity of manipulating vitamin D3 levels in beef feedlot cattle and altering these same pathways that we've been talking about this morning, specifically the same pathways that are related to steroidal implant mode of action. The receptor for vitamin D3 is actually very similar in structure, and it is part of a superfamily of steroid hormone receptors. So in fact, we think of vitamin D as a very crucial essential nutrient, but in reality, when vitamin D binds to its receptor, it's activating pathways that are very similar to estradiol binding to the estrogen receptor alpha, or trenbolone 17 beta binding to androgen receptor in skeletal muscle tissues. And so to lead off with that, we're going to give some examples. First, we're going to start with some examples with poultry, with avian species, and some early work done showing the effect of high D, or this would be 25 hydroxycholocalciferol or 25 hydroxy vitamin D3 supplementation on skeletal muscle growth and satellite cell activity in broiler chicks. And this work was done, I want to give a shout out to one of my early graduate students in my career at Kansas State University, Dr. Jessica Starkey, who's done great work on the poultry nutrition and growth side at Auburn University. What Dr. Starkey's group showed is that by feeding very low levels, we're talking part per billion levels of high D, 70 micrograms per kilogram of feed, so 70 parts per billion concentration of added 25 hydroxy vitamin D3, Dr. Starkey's lab showed an increase in the activity of these satellite cells that I talked about early on in my webinar. In fact, these cells then ultimately fused into the existing muscle fibers and brought about a larger cross-sectional area in the psoas major of these broiler chicks. Another lab group did very similar work with the same compound, 25 hydroxycholocalciferol in broilers. And interestingly, they took a different approach looking at protein synthesis and protein degradation, and they saw changes in this mTOR pathway that I talked about early on that is a key regulator of stimulating protein synthesis and reducing protein degradation. So again, by feeding high D to these chicks, they showed changes, improvements in protein synthesis, and this was related again to changes in the mTOR pathway that has been shown to be so critical at affecting protein synthesis. So you might be thinking, well, this is fine and good that we can feed a product like high D or 25 hydroxy vitamin D3 to birds and see these differences. But really, what happens in larger species or in ruminant species like beef cattle? And a few years ago, a study was done down in Brazil. And just to kind of set the stage here, these were intact bulls. These were Nelore bulls. So they had high levels of testosterone. Of course, in Brazil, implants are not allowed to be used, nor are beta ligands allowed to be fed. So these are really natural cattle, but intact males with high levels of testosterone. This work was done at the University of Vanuatu, and I want to again highlight the senior scientist, Dr. Mario Aragoni. Dr. Aragoni has trained so many feedlot nutritionists in Brazil. He's a great scientist, a great researcher. Another co-author on this manuscript, Dr. Danilo Milan, is currently a professor in Sao Paulo State, and Victor Carvalho was a representative from DSM that was also involved in this research of, again, looking at 25-hydroxy vitamin D on growth parameters in Nelore bulls. So some take-home messages from this work. Again, looking at the doses that were fed, they had a negative control, and then they had 2 doses of high D, 1 milligram per head per day or 3 milligrams per head per day. So very, very low levels when you think about dosage of this naturally, or this natural alternative vitamin D3 compound. So what were some observations that the group in Brazil saw? Remember all the things I talked about with IGF-1 and the mTOR system. You can see that, in fact, both 1 and 3 milligrams of high D caused an increase in IGF-1 expression in muscle, and that in turn led to greater changes in this mTOR pathway, which again is going to help support protein synthesis and reduce protein degradation. So from a big picture standpoint, you can see that by feeding the high D product to the Nelore bulls, we saw a numerical increase of almost 9 pounds of hot carcass weight with a 1 milligram feeding of high D to these bulls. We had significant increase in dressing percent and a tendency for an improvement in longismus or ribeye area, longismus muscle area. So back from the previous slide, we knew that the high D affected IGF-1, affected the mTOR pathway. The net effect of that was a numerical 8 to 9 pound increase in hot carcass weight. And so again, these animals were not implanted, but they were intact males. So what I find exciting is that I would expect that in the United States, under our now normal implant regimens, that again, the use of a product like 25-hydroxycholecalciferol could lead to something like 8 or 10 pound improvement in hot carcass weight, which in today's market would be worth $35 to roughly $40 a head by increasing hot carcass weight an additional 8 to 10 pounds. The last area that I want to talk about has to do with another vitamin, vitamin A. And we've done a lot of work in our lab over the years of looking at some of the active ingredients of vitamin A once it's absorbed through the GI tract of cattle. And in circulation, we can measure retinol, and retinoic acid. And specifically, a lot of work has been done with retinoic acid, showing that high retinoic acid in a cell culture model actually inhibits marbling development of preadipocytes, of intramuscular preadipocytes that ultimately will go on to fill with lipid and become marbling. And so that's caused some concern that the relationship of higher vitamin A levels potentially can reduce marbling. But in fact, what's kind of neat and almost contradictory to this is that some of the work we did, and from one of my former PhD students, Dr. Jungju Kim, who's now a professor at Michigan State University, we showed that retinoic acid actually impacts this myosin heavy chain expression that I've talked about earlier. And in many ways, in a positive way for meat quality, that higher levels of retinoic acid actually cause an increase in myosin heavy chain type 1, which we need for more marbling, right? And leads to a more tender product. When we have higher levels of retinoic acid, we actually reduce the myosin heavy chain type 2X. And so this has brought about by a very important transcription factor in muscle called PPAR delta. And what JK showed is that higher levels of retinoic acid stimulated PPAR delta in such a way that we turned on the expression of myosin heavy chain type 1. So I just all morning have talked about if we want more marbling, we have to have more myosin heavy chain type 1. So we've set the table early on. I think we need vitamin A. It's critical at getting the right fibers in the mix to have marbling. But it's almost like then if there was a way we could block that towards the end of the feeding period and allow those adipocytes associated with the type 1 fibers to fill with lipid, we could actually improve marbling by kind of phase feeding different levels of vitamin A. And we definitely need a lot more research in this particular area. But I know as a nutritionist, the last few years, you've maybe heard about the negative aspects of high vitamin A to marbling. But in fact, we need the vitamin A early in the feeding period to really set the table to get the right fibers in place to have that marbling later in the feeding period. So it's not as easy as just yes or no. We have to look at this at a much deeper level to kind of understand the mode of action of this. So to kind of conclude this morning, some major take-home points. We talked about two major growth promotants that we've used in the United States for several years to support more efficient muscle growth of our feedlot cattle. Anabolic steroids, the active ingredients in steroidal implants, and the orally active beta ligands or beta adrenergic agonists. We talked about how beta agonists can impact this very important enzyme that controls energy metabolism in skeletal muscle, the AMPK pathway. And in fact, there may be other alternatives that can perturb that AMPK pathway in a positive way to support more efficient postnatal muscle hypertrophy. I talked about the role of these satellite cells, all the things we've learned by using anabolic steroids in cattle. We've learned how satellite cells support postnatal muscle hypertrophy. But both in poultry and in the one beef study conducted in Brazil, we've now shown that a product like 25-hydroxyvitamin D3 or high D can also increase satellite cell activity and also support the mTOR pathway at causing an increase in protein synthesis. In addition, we talked about other vitamins. I talked about biotin potentially impacting AMPK pathway from the first bullet, but also the importance of understanding things like vitamin A or retinol, and retinoic acid impact on muscle fiber hypertrophy and how that relates to meat quality or marbling and tenderness. So obviously, there's more than one road or one formula to get to the same endpoint as this final slide kind of illustrates, right? We've talked about using growth promotants, steroidal implants, and beta ligands for so many years in the US. But we can use that to increase our understanding of the mechanism of action of postnatal muscle hypertrophy, and in fact find alternatives, things like 25-hydroxyvitamin D3 or biotin or altering vitamin A. These alternative mechanisms or alternative molecules are going to work through the same mechanism. So they're going to get us to the same endpoint, right? Or they're going to be complementary or maybe additive to some of our existing exogenous growth promotants like implants and beta ligands. Remember what I said about beta ligands will not cause an increase in satellite cell activity the last 30 or 56 days. So we have to set the table before then to make sure we have adequate nuclei in those fibers. We showed how something like 25-hydroxyvitamin D3 can actually cause an increase in activation of satellite cells. We can be feeding this prior to the beta ligand to ensure additional nuclei are within that fiber to get the most out of the beta ligand at the end of the feeding period. So again, it's been a great privilege to present this morning to this very important topic. of ways to support muscle growth through alternative mechanisms. This time I'd be happy to address any questions. Thank you very much.
Thank you, Dr. Johnson. This was a great presentation on ways to manipulate muscle growth. As a reminder to our audience, you can still submit questions. We've got a few minutes for that. I also would like to remind the attendees this morning, since we are having a global discussion with multiple attendees from different regions in the world, to please be in contact with your dsm-firmenich representative regarding the availability of a product such as Heidi. It is available for inclusion in cattle diets in the US. But please confirm with your representative about availability in your specific region. I think with that, we'll address a few of the questions we've received, Dr. Johnson. One of the questions we received was, what type of response would you expect when comparing the impact of growth implants versus 25-hydroxy-D3 on impacting muscle growth and having that effect on IGF-1?
That's a great question, and that's kind of what I ended with. I have not. So the only bovine data I really have access to with a product like Heidi would be the study done in Brazil, and of course those were intact bulls with no exogenous growth promoting tools. And they saw a numerical 8 to 9 pound increase in hot carcass weight. Obviously, in a different rearing situation, lighter animals fed less days on feed. So this is a big leap, but if we could equate kind of our standard current implant protocols in the grow finishing phase under the new guidelines of guidance 191, we know we might get a 25 to 30% increase in average daily gain compared to a non-implanted steer with an implant. I really think that by incorporating the Heidi throughout the entire feeding period with the TBAE2 implant program, with our longer fed cattle in the US production situation, I really think we could get somewhere from 5 to 10 pounds additional hot carcass weight, matching up with what they saw in Brazil in animals that had really high levels of testosterone. But we know testosterone is going to stimulate satellite cells, going to stimulate the mTOR pathway. Again, we don't have the data in the US probably yet, but I do think we should expect maybe somewhere between 5 and 10 pounds additional hot carcass weight on top of what we would expect from the implant.
That makes sense. I believe we've reached the end of our time for this webinar. If there's any other additional questions, please reach out either to myself or Dr. Johnson, and we can address any of the questions you might have. I'd like to thank everyone for joining this webinar this morning. At dsm-firmenich, we do aim to provide thought leadership in the industry to further sustainable, profitable beef production as well. We look forward to joining you on another webinar in the future and talk about beef production. So with that, I say thank you, Dr. Johnson, and thank you to everyone for joining us this morning.